How is it that we find ourselves surrounded by such complexity, such elegance?
The genes of you and me, the genes of you and me, are all made of DNA. We're all made of the same chemical DNA. We're all made of DNA.
Hi, you're listening to DNA Today, a podcast and radio show where we discover new advances in the world of genetics. From genetic technology like CRISPR to rare diseases to new research, we have you covered. For a decade, DNA Today has brought you the voices of leaders in genetics. I'm Keira Deneen. I'm a certified genetic counselor and your host. Today we're diving into FSHD, which is a muscular dystrophy, and highlighting the utilization of whole genome mapping in the diagnosis process. Joining me to explore these topics are June Kinoshita, who is the Director of Research and Patient Engagement at the FSHD Society, and Rojan Kavosh, who is a genetic counselor by training and is currently a genomic testing consultant at PerkinElmer Genomics. I wanted to start out, June, by picking your brain a little bit and setting the scene on what FSHD stands for and what muscles in the body it primarily affects, since this is a muscular dystrophy.
Sure. So it's a mouthful, I'm going to warn you. It's Facioscapulohumeral Muscular Dystrophy. You can see why we call it FSHD for short.
Definitely.
But as its name implies, it typically affects muscles in the face, the scapula, which would be like the shoulder blades, the humerus, the upper arm. But it's actually a misnomer because muscles throughout the body can be affected, and stereotypically it would be in addition to the face, shoulders, and upper arms, it can be the abdominal core muscles and hip girdle, the tibialis anterior, which is the sort of front of your shin, and if that's weak, it causes a foot drop, and people experience that often as a very early symptom in FSHD. The hamstring can also be affected, but people don't often realize it because there's, 4 gigantic muscles in your thigh, and so one goes, but the others are there to support it. But that's very common in people with FSHD. The really unusual thing about FSHD is that the muscles are not affected all at the same time in the same way. They will seem perfectly healthy, and then one of the muscles will start to degenerate. And then a little while later, another one might degenerate. It's often asymmetrical in onset. That's kind of a signature, like a real telltale. If you lose one bicep and not the other, you might suspect that you have FSHD.
Interesting, because a lot of genetic disorders, you're looking for more bilateral things affected where you have more symmetry there. So that's, an exception, I would say, in terms of genetic conditions. When do these symptoms usually start appearing in people, or is it a wide range?
It is a wide range. We have infants who are born with poor muscle tone who turn out to have FSHD, and those kids might be in a wheelchair by the age of 10. And then you have senior citizens who don't even know they have it, and then one of their adult children is diagnosed, and then when they test the grandparent, it turns out they've had it all along, but they're so mildly affected that nobody ever picked it up. So it is both a wide range. Very typically, though, about 80% of males are diagnosed during their adolescent years. So there seems to be an acceleration or an onset of the muscle degeneration associated with time of puberty. Women seem to develop it a little bit later. But it is challenging because there's no cookie cutter pattern. It's a wide range. But that's a clue genetically in terms of what's involved in this disease.
And I'd imagine that because it's hard to diagnose in this way, because there is so many different symptoms, but also different ages of onset, that it's vastly underdiagnosed.
Absolutely. Very much so. And I think the average patient journey for a condition like this, and certainly with FSHD, is about 10 years from the first onset of symptoms until you finally walk into a doctor's office and the doctor says that's what it is. Very much so.
Yeah, that's certainly very challenging. And I think it's hard because at one point it was considered to be like an ultra or nano rare disease, but how common is it estimated to be today now that we have more information?
So the estimates range between about 1 in 20 or 1 in 15,000 to as lower than one, less than, more than 1 in 8,000. That last number is from a Dutch study, which used a very rigorous method, and they were able to capture a large percentage of people with clinically diagnosed FSHD within the Netherlands. So the methodology was very strong, but the caveat is it was, in what might be a slightly genetically unique population. But even that number they thought was an underestimate, because they said a lot of people, this is familial, it's actually transmitted in an autosomal dominant way, at least FSHD type 1 is. So if it's in the family and then people in the relatives see similar symptoms, they may kind of suspect that that person, cousin so and so has it and grandma has it, but those individuals may not necessarily go and get a clinical diagnosis. They just kind of live with it. So the Dutch investigators said they were sure that this was an undercount based on what they knew about their community.
Which is certainly, I think, a common theme with like rare diseases that we see. Oh, it's actually more common than we originally thought. I feel like I hear that from so many different people and patient advocates. And as you were kind of alluding to there's two types of FSHD. Are there differences in symptoms between the two types? We're going to get into the whole genetic side too. But are there differences in symptoms between them?
Generally, the folks who are studying this say there are not. But, we're talking fairly small numbers. And there was a French study a few years ago that seemed to suggest that maybe people with FSHD2 might have a higher prevalence of skin conditions. I don't know if that study's been replicated, but more and more is coming out all the time. But for general purposes, we'd say they are pretty much indistinguishable clinically. And one of the reasons for thinking this also is that they both genetic mechanisms for FSHD type 1 and type 2 converge on the same sort of pathogenic pathway, which is this expression of this gene called DUX4, which is normally not expressed in the adult muscle. And both types 1 and type 2 result in the breakdown of a mechanism that keeps DUX4 locked down and unexpressed. So that mechanism breaks down, DUX4 gets expressed in both types of FSHD, and the disease is thought to result from the downstream toxic consequences of DUX4 expression.
So it's the expression that's a problem in healthy, typical people. They don't have that expression once they hit adulthood. Am I getting that right?
Correct.
Okay, interesting. So just to bring you into the conversation a little bit, are most cases inherited from an infected parent, as we've been talking about with June, or is it usually random mutations, like what we call de novo mutations in people that are diagnosed?
Yeah, most individuals, about 70%, are diagnosed with FSHD and have inherited it from an affected parent. About 30% of cases are de novo or come from a random mutation.
And when I was looking a little bit into this, it was interesting. Something came up with type 2 called digenic inheritance pattern. I got to be honest, I'd never heard of this before. So can you explain what this type of inheritance pattern means? And, as we've been talking about, type 2 is less common than type 1. But I think it's interesting just to look at the genetics and the differences there.
Yeah, that's right. So type 2 accounts for about 5% of cases of FSHD compared to type 1, which accounts for about 95% of cases. Digenic inheritance means there are two alleles with pathogenic changes that interact to cause a genetic condition. So we'll get into this a little bit more, but FSHD type 2 results from double heterozygosity for both an FSHD permissive DUX4 allele, what we call haplotype 4QA, and a pathogenic variant in the SMCHD1 gene.
So it's that combination that then results in having the disorder and having the symptoms from that.
Right.
And as we were talking about before, people in even one family that have like the same mutations can have different symptoms, which I always find is interesting. Like your genetics could be the same for that, but one person could have, certain muscles affected and another person had different muscles or a different level of severity with that. HemoShare Therapeutics is dedicated to developing therapies that help improve the lives of patients with rare genetic diseases. HemoShare scientists have developed an investigational therapy to treat methylmalonic acidemia and propionic acidemia. These ultra-rare genetic diseases lead to the rapid buildup of toxins in the body. Patients can suffer from intellectual disability, liver, kidney, or cardiac disease, and other life-threatening conditions. HemoShare is conducting a clinical trial to assess if their drug may be effective in the treatment of both methylmalonic acidemia and propionic acidemia. Learn more about HemoShare and these ultra-rare metabolic diseases in episode 188 of DNA Today. You can also visit hemoshare.com. That's HemoShear.com. June, what are the testing options for people that are suspected to possibly have this disorder? Like, what's typically the first test ordered, I suppose?
I think, well, if they have a clinical workup and the clinician thinks this looks like FSHD, I think the first test ordered would probably be for FSHD type 1. And there are 2 labs in the US that currently offer it, 2 large labs. There's a few small ones that I don't even know, I'm not sure if they're still around, but Perkin Elmer Genomics is one of them, and the University of Iowa is the other one. And so they would be testing for what's called here we get into the actual genetic mechanism, and it's
let's nerd out about it.
It's a bear. It's a good one to test if you teach genetics. That's the bonus. This is going to be the bonus question.
I was going to say extra credit for people that can answer this and follow along, right?
Exactly. So the I'm not even sure mutation is the right word, because it does not consist of a single nucleotide polymorphism, sort of garden variety mutation. This change in the genetics or the genome occurs at the tip of chromosome 4, the long arm of chromosome 4. There's a region that is filled with these what are called D4Z4 repeat units. Each unit is about 3 kilodaltons, I think. Correct me, Rojan, I don't know if I get that mixed up, but they're repeated like beads on a string. And your typical person, we all have this D4Z4 repeat region on chromosome 4, and you might have anywhere between like 10 to 100 or more of these repeats. If you have fewer than 10, but you have to still have at least 1, so between 1 and 10 of these repeats, you are going to be at increased risk of developing FSHD. You also have to have, in combination with that contraction of D4Z4, what Rojan referred to earlier as a permissive haplotype, polyQA. So this is a polyadenylation signal so if I back up a minute, D4Z4, they contain this DUX4 gene, which I'd love to talk about later because it is a really amazing gene. But it's in this region, and normally the DUX4, it's called it was described as almost a pseudo gene or in early days, because it wasn't stable on its own. It just would degrade. But in the presence of the 4QA polyadenylation site, that would stabilize the RNA and the DUX4 could be expressed. So you need both the contraction and the 4QA haplotype. People also have a 4QB, which is not permissive. It doesn't stabilize DUX4. And if you have the contraction but you have 4QB, you are not at risk of getting FSHD. So it's pretty complicated.
It's definitely very complicated. And I think there's a reason why genetic counselors would be really helpful in this diagnosis when people are finding out about this, so that they can understand all of, the genetics behind it, or at least enough to understand, like, this is how the symptoms happen, and like, this is what the risks are for other family members. And interesting that, usually for genetic disorders, like I think like Huntington disease, other disorders, you have an extra repeats, but for this you actually have less than an average person, is what it sounds like, that leads to the disorder. That's really interesting, and kind of a lot of exceptions that I'm learning about FSHD. So can you tell me a little bit more about how genome optical mapping fits into this, and maybe giving us like a baseline explanation before we apply it to FSHD?
Yeah, so whole genome optical mapping is a process where we isolate large fragments of DNA, so upwards of 150,000 base pairs, from a whole blood sample. And for reference, typical next generation sequencing uses very small DNA fragments of about 100 to 200 base pairs. So these are much larger pieces of DNA. And saliva swabs and even older blood samples are not ideal for getting this size of DNA fragment, since DNA denatures over time when it's outside of the body. So those would not be good sample types for optical mapping. Then fluorescent probes are applied to these large DNA fragments, arranged every 6 base pairs or so. So that's about 500 million probes per genome. And another reference point, a microarray, commonly ordered genetic test, uses a few 100,000 to a couple million probes. So this is many, many more than that.
So it's almost like a halfway point, I'm thinking, between like a microarray and like doing a whole genome sequencing. Like you're kind of in the middle of that in terms of like you're looking at really big chunks of bigger chunks of the genome than if you were doing sequencing where you're looking at every single letter.
Right. So this is different than even traditional whole genome sequencing or microarray, because we're not sequencing through everything and looking for single nucleotide variants or copy number variants like we would usually do, but we're rather specifically looking for the haplotype that June mentioned and D4Z4 region sizes, which have implications for FSHD. So those large DNA fragments labeled with their probes are passed through very tiny tubes in an analyzer, tiny enough where only a single strand of DNA can fit into each tube to be read at a time. And this way they are able to be linearized and we get high resolution images of each strand. And then lastly, we use a process called de novo gene mapping, so that means without using a reference genome, to visualize a haplotype, whether it's A or B, the repeat number of the D4Z4 region with an approximately 1 repeat size, and any complex or balanced rearrangements.
So because the repeat size is so big, you're able to say, okay, like how many of those repeats are present? That's part of the process.
Correct, yes.
Very interesting. So then, I think we've talked a little bit about the genetic etiology of the condition in general. Diving a little bit deeper, what is the genetic difference, if there is one, between the two types? Because if you're doing testing, we talked about like, all right, if you're suspecting the disorder, you're going to try to test for type 1 first, because 95% of people with the disorder have type 1. So what is the genetic difference between the two types?
Yeah, so just to back up a little bit and reiterate kind of what June said earlier, DUX4 is the gene that is turned on or activated in both types of FSHD. And it's located in that D4Z4 repeat region of chromosome 4. And like June said, we've learned that we need DUX4 to be turned on during very early stages of life, but when it remains turned on all the time, that protein product of DUX4 is toxic to muscle cells. And our cells have different ways of turning off genes that don't need to be activated all the time and in all the different cell types in our body. One way they do this is by methylation. And this chemical marker, when it's attached to the gene, it tells the cells to keep the gene off so the protein product can't be made. And if methylation doesn't occur, we call this hypomethylation, the gene is not being turned off as it should. So in FSHD type 1, DUX4 stays turned on because there are a fewer number of D4Z4 repeats, specifically less than 11, so between 1 and 10. So methylation cannot occur to turn off the gene because of this D4Z4 contraction. In FSHD type 2, there is a normal number of the D4Z4 repeats, but there is a pathogenic change in the SMCHD1 gene that inhibits its proper protein production, which in turn interferes with methylation, so again, methylation can't occur. But for either type of FSHD to occur, the haplotype 4QA must be present on the chromosome. And a haplotype is a pattern of specific variants of genes that tend to be inherited together. And as I had mentioned earlier, the FSHD permissive DUX4 allele that needs to be present is the haplotype 4QA that we've referred to.
So it's really two different ways of the DUX4 gene not being methylated, so that it's not being shut down, and so then it's being expressed. So there's just the two different ways for type 1 and 2. So just interesting that that's how you can figure it out, and just that, when I hear haplotype, like one of the first things I think of is like ancestry. Like we're looking at groups that are being, groups of genes that are being inherited together. And that's kind of how some people can track ancestry. So that makes sense of looking at, okay, this is inherited together with that. So as a genetic counselor by training, I mean, when do you recommend for genetic testing to be ordered for an individual? I imagine sometimes that's when someone's symptomatic, but is there like an age recommendation, or if there's potential people in the family that could be affected? Like, what's your view with that?
Yeah, I think, like June said, it'll come down to when it is a clinical suspicion that there could be FSHD going on. Typically, the people who should consider genetic testing should be, somebody who either has or their child has a suspicion to have a muscular dystrophy and they haven't had genetic testing, or if their clinical diagnosis that they currently have doesn't seem to fully explain the symptoms, or if previous genetic testing didn't provide a diagnosis, or if you have a family member diagnosed with FSHD, then testing can be considered. But FSHD can have overlapping symptoms with other neuromuscular conditions, and sometimes a diagnosis can be difficult to distinguish. So the provider, would have to make that decision to include testing potentially for a larger panel of potentially associated genes in addition to FSHD 1 and 2 testing, or to start with those two and then, pursue additional testing if needed.
Yeah, certainly. Because I would think, as providers, you wouldn't necessarily have it's tough to say, oh, I think it's this specific muscular dystrophy. I think some of them are very unique, and you can say, okay, we really think it's this one, or depending on like carrier screening or other genetic information that you have. Hemashire Therapeutics is dedicated to developing therapies that help improve the lives of people with rare genetic diseases. Hemashire is currently conducting a clinical trial for a potential new treatment for 2 ultra-rare genetic metabolic diseases called propionic acidemia and methylmalonic acidemia. Learn more about Hemashire and these ultra-rare metabolic diseases in episode 188 of DNA Today. You can also visit hemashire.com. That's HEMOSHEAR.com. We talked about Perkin Elmer Genomics being one option for ordering this. What are some of the benefits of ordering through Perkin Elmer specifically for FSHD, or, looking at the panels that Perkin Elmer offers for muscular dystrophy too?
Mm-hmm. Well, traditionally, testing for FSHD has included complicated algorithms. It would typically start with Southern blot to estimate the D4 to Z4 repeat sizes, then followed by haplotyping, then methylation studies, and then finally Sanger sequencing or next generation sequencing of the SMCHD1 gene if that's needed. And from a clinical standpoint, multi-step testing approaches like this could be complicated, could be costly, and of course time consuming. So Perkin Elmer Genomics has adopted a new approach to FSHD testing, optical mapping, as we've discussed, and that makes this process less complicated and more affordable as well. And if there is a provider who is also considering additional testing options, we have some panels on our test menu, such as our 132 gene comprehensive neuromuscular disorders panel that uses next generation sequencing to read through the panel of genes the same way that we would for the SMCHD1 gene, so it creates this efficient way to look at multiple genes at once without any changes to that turnaround time.
Yeah, so that's very helpful because, as June was bringing up earlier, it can take patients like average 10 years, I think you said, June, of having a diagnosis for FSHD. So, any ways that we can speed that up is really just going to help patients. June, how have you seen patients benefit from genetic testing, of just the patients and patient advocates that you've met with?
I think, I've heard so many stories from people who for 10 years or more, many years, just didn't know what was wrong with them. They were told they were imagining these issues, etc. They were thinking maybe they got an injury. Some of them had surgeries that they didn't really need. And then suddenly when they got the genetic diagnosis, they suddenly said, okay, I know what I have now. I know what I am. And that the incredible relief, there's a certain kind of, it's not just this open mystery anymore. There's this immense sense of relief that they have an explanation. And I think in the case of FSHD, because we have such a strong dynamic patient advocacy community when they find that. I mean, if they didn't have the diagnosis, they wouldn't know who to turn to, who to connect with, what to do, how to be proactive, even know what's best in terms of their health care. One thing that's really important, again, one of the really vexing, difficult things about FSHD is you can have somebody who's virtually non-symptomatic, who carries the mechanism for FSHD, pass it on to a child who then, for whatever reason, it might be environmental, it might be another modifying gene, develops a much more severe form of the disease. And the parent didn't know, and they bear this burden of guilt, which they shouldn't, because they didn't know, they had no way to know, but they still do. That's human nature. And so it used to be that you might even have FSHD in your family, but if you didn't have symptoms, you were assumed to not be affected. And people go ahead and start families and discover they, in fact, were carrying the gene. And so I think genetic counseling practice has had to keep up with this, that being asymptomatic does not rule you out if you have first-degree family members who are affected. Many people have, maybe the parents are no longer around, they can't test the parents, but they have a sibling who's affected. Well, then they are, if you can't test the parent, you should be tested yourself. So it has all kinds of ripple effects and implications throughout the family.
Yeah, definitely. And I would think that having a genetic diagnosis would also open up the door for some clinical trials that I imagine are starting. Are there any clinical trials that you're aware of now that patients can enroll in or learn a little bit more about and become partners in research?
Absolutely. This is a really exciting time in FSHD. Because this DUX4 gene is actually a great therapeutic target, because if you can shut down its expression, and there are multiple ways you could try to do that, you're not going to have toxic effects, because that gene does not have a life-sustaining role in the adult, right? As far as we know, if we looked in many tissues, it doesn't seem to. So it's a great therapeutic target. And there will be a phase 3 trial of a drug, a small molecule drug that shuts down DUX4 expression, starting up very soon, probably within the next 2 or 3 months, maybe even sooner. And that is a phase 3, recruiting about 240 patients. So that's something that you would potentially qualify for, but you would need to know that you genetically, have FSHD. There are other trials that are being planned that might be starting up late this year or early next year. It's a growing area. Every few months, another company enters the space. So we want our community to be empowered with the knowledge to, get the testing they need and to know how to connect with these opportunities to participate in research.
Yeah, as you said, the first step would be having that genetic diagnosis so that it does open up the doors for everything you're talking about. Even I think just having an answer, just from talking to a lot of patient advocates on this show, like that was just so empowering to know, yes, there is a genetic change that has led to these symptoms. And I have an understanding for that now. And, I think a lot of patients are told, well, maybe it's more psychological. And sometimes for some people it may be, but for these disorders, that's not the case. So I think that's an important part, and very exciting to hear that, you're that close to phase 3 of the clinical trial and everything. Do you have a prediction on what year treatment may become available, looking at just the scope of things, or am I just dreaming to have an answer there?
Well, no, we've made an educated guess. I mean, we went a little bit out on a limb, but we think, given everything we're seeing, that there could be the first approved drug on the market by 2025. So that's only 3 years from now.
That's very exciting. Yeah, that's within reach, I feel like. That's not too far away. And as we've explored on this show in so many episodes, genetics moves so fast, and that is just such a great thing, because it's just helping so many more people, and it makes sense genetically too, that if we can just shut down and methylate this gene, then we're not going to have the toxic effects and the symptoms that we see from this. So as you said, like, it is a good target. Like, I've been reading a lot of books about CRISPR recently, and those are the ones that they say, oh, this is easier, rather than like fixing a gene, just shut it down and then that person doesn't have the disorder. Those are higher on the list of like, these are going to be easier to treat and kind of, I don't know, I kind of want to use the word cure there, but I'll hold off on really putting that out there yet. But anything else to mention before we wrap up the show? I mean, I have just learned so much. I think it's just amazing how, as much as I learn about genetics, there's just always so much more to learn.
Well, I do want to make a pitch for natural history studies because they don't get the emphasis that they deserve. No clinical trial is possible without the natural history data. And of course, again, having a genotypically defined population that you're studying somebody with FSHD, not something that just looks like it, is so important. And there's a study going on now called the MOVE study and MOVE Plus, which is based on a visit to your doctor. I mean, it's got to be at one of the sites that's participating in the trial. There are now, I think, 12 in the US. But it's the largest natural history study being done for FSHD. And the success...
Wow. We'll definitely link to that in the show notes then so people can check it out.
Absolutely. And I don't know if we're out of time. If the audience of geneticists, I just want to say DUX4 is an amazing gene. When I started being involved in this field, nobody knew what it was. I looked up PubMed. There were maybe 4 papers about DUX4. It was this weird double homeobox gene with no known function. And a few years ago, it was discovered that DUX4 becomes expressed in, I think, maybe day 2 of human development. In the fertilized egg, as it's dividing, it's one of the first genes that gets turned on. And it appears to be the master gene that sets in motion the whole genetic program of human development. So
we should study it just for that.
It will be everybody.
We're going to learn so much.
Exactly.
Wow.
Everybody will know what DUX4 is who studies genetics.
Yes. All right. Now I can say I know what DUX4 is. So that's helpful. Well, thank you so much both for coming on the show. And I think it's just so interesting to be able to hear both of your perspectives too, coming from the FSHD Society and PerkinElmer Genomics. I think that's just really helpful to look at both sides, and coming together really is one and everything. So I really appreciate just teaching me and my audience about FSHD and all the interesting genetics that play a role with this. But thank you so much for joining us.
Thank you.
Thank you. I think it's so cool. You have a whole show dedicated to DNA today.
Totally. Yeah.
I'm that nerdy. I just can't stop talking about genetics. So, for more information about today's episode, visit DNApodcast.com, where you can also stream all episodes of the show. We encourage your questions, comments, guest pitches, and ideas. Send them all into info@DNApodcast.com. Search DNA Today on Twitter, Instagram, YouTube, Facebook, so you can connect with us there. And a favor, please rate and review the podcast on Apple, Spotify, or wherever you listen. This truly helps us climb the charts and allow more genetic nerds like yourself to find the show. DNA Today is hosted and produced by myself, Keir Deneen. Our social media lead is Corinne Merlino. Our video lead is Amanda Andrioli. Thanks for listening, and join us next time to discover new advances in the world of genetics.
The genes of you and me, the genes of you and we're all made of DNA. We're all made of the same chemical DNA.